Hello dear students, I'm Assistant Professor V.S.Aher, and in our today's session we are going to study about bacterial genetics, in which we are going to study in detail about the chromosomal and extra-chromosomal material present inside the bacterial cell. So before studying about the bacterial genetics, one must know what is genetics. Now the genetics is a branch which includes the study of genes, their structure, functions, their hereditary and variations carried out in the genetic sequences, in which there are various terms like genomics, which includes the study and analysis of nucleotides of DNA. As whenever we consider about the genetic sequencing, one must know about the nucleic acids. In previous sessions we have already studied about the different types of nucleic acids, in which we have studied about the various structural and functional components about the nucleic acid, which included DNA as well as RNA. So we have already seen that the Watson and Crick, for the first time, they have studied the structure of DNA in the year 1857, and they have found that the complex structure of DNA is double helix, which includes different types of base pairing. If we consider about DNA, now the DNA, it stands for deoxyribose nucleic acid, and this DNA, it is composed of two types of nitrogen bases, which includes purines and pyrimidines, in which purine is adenine and thymine, and the pyrimidine group present inside the DNA molecule is cytosine and guanine. Now the ratio of these nitrogen bases, it varies inside the DNA molecule depending on the species. Here is the structure of DNA. Let's study it in detail. Now a DNA molecule, it is composed of two chains of nucleotides. Now nucleotide includes nitrogen bases, phosphate group, and sugar molecule, and these nucleotides are joined to each other with the help of phosphodiester bond, and a long chain of nucleotide binds to the another nucleotide chain, and together they bound together to form a double helix structure, and this double helix structure, it is known as deoxyribose nucleic acid. And each chain, it consists of deoxyribose sugar, phosphate group, which are arranged alternatively. See, here you can see the sugar phosphate backbone is present, as well as different types of base pairing of adenine and guanine. Sorry, different types of base pairs are joined. Adenine is joined with the help of hydrogen bond to thymine, and cytosine, it joins to the guanine with the help of hydrogen bond. Now if we consider the structure of DNA, it is attached to each other alternatively and the distance between two successive bases is near about 3.4 Armstrong unit. This structure we have already studied in detail in the previous sessions. Then the second type of genetic material or the chromosomal material present inside the bacterial cell is the RNA. Now RNA is the ribose nucleic acid. If we study the difference between DNA and RNA, there are three major types of differences. First one is the difference between the sugar molecule. RNA consists of ribose sugar, whereas DNA molecule it consists of deoxyribose sugar. There is only the difference of one oxygen molecule. In the second difference is in the pyrimidine group. The pyrimidine present in the DNA molecule includes sorry. The second difference is in the purine group, which includes uracil in the RNA instead of thymine. And these RNA are made up of different types, which includes mRNA, which is messenger RNA, and the mRNA plays an important role in the transfer of messages during the protein synthesis. Ribosomal RNA it plays the important role in the formation of ribosome during protein synthesis. And the third type is transfer RNA. The transfer RNA is plays an important role in the transformation of amino acids during the protein synthesis. Now here you can see the structure of both DNA and RNA. The structure of DNA is double helix or double stranded, and this is the third important difference between the DNA and RNA. The structure of DNA is double stranded, whereas the structure of RNA it is single stranded. Now if we consider about the molecular biology, we have seen the central dogma of life, in which DNA is very important, which carries the genetic information from one generation to another generation. When this DNA is transcribed to RNA, there is a formation of polypeptide chain, and this process is known as transcription process. And the next process in the central dogma was the formation of protein or the synthesis of protein from the RNA molecule, which is known as translation process. Now these cell function it depends on the specific polypeptide and specific types of enzymes. Generally, DNA is present in all the different types of organisms in prokaryotes as well as in eukaryotes as a genetic material, excluding viruses which contains DNA or RNA as their genetic material. So in genetic or the bacterial genetics, the most important term is genes. Now genes are nothing but fragments of DNA. It is a sequence of DNA which carries codons specifying for particulate polypeptides. It may contains a combination of different types of nucleotides, and generally the codons are made up of three types of amino acids. Then if we consider about the bacterial chromosome, which is our actual part, this bacterial chromosome it contains double stranded molecule of DNA arranged in a circular form. sorry, the DNA which is present inside the bacterial chromosome, it is present in the circular form and it is double stranded, and its length is near about 1000 microns in case of bacteria. And bacteria DNA contains about 4000 kilobases. 1 kilobase includes 1000 base pairing. Then it may be of adenine, thymine or guanine and cytosine. 1 kb मतलब 1000 बेस पेयर होता है, तो बैक्टीरियल डीएनए में 4000 किलो बेसिस प्रेजेंट होते. सो वी कैन से दैट 4000 into 1000 means it includes near about 40 lakhs of base pairs in a single DNA molecule. And if we consider about human beings have near about 3000 kilo base pairs. So this was all about the chromosomal part of bacterial cell, which includes nucleic acids like RNA and DNA. The next important part is extra-chromosomal genetic material. Now, bacteria, it generally possesses extra-chromosomal genetic material, which is generally not essential for the survival of bacteria, but it plays a vital role in the bacterial cell, such as resistant to different types of antibiotics, or it can also produce different types of toxins. Now, the first and the most important extra chromosomal genetic material is the plasmid. The plasmids are extra chromosomal, circular, double stranded DNA molecule present in many species of eubacteria and archaebacteria. These are about 1/10 size of bacterial chromosome and have about 5 to 100 genes present in it. Now, different types of plasmid can be found in a single species, and generally they are present in the independent form inside the cell, as well as they multiply independently from the cell reproduction. The cell reproduction does not forms any impact on the multiplication of the plasmids. These plasmids have the most common vectors in the recombinant DNA technology and have found various applications in the industrial technologies using genetic engineering. These plasmids can also be present in the various different types of cells like yeast cells, and certain plasmids, they may occur in a single copy per cell, while other may be present at the concentration of 40 or more per host cell. मतलब एक सेल में एक सिंगल प्लाज्मिड भी प्रेजेंट हो सकता है या फिर मल्टीपल नंबर ऑफ प्लाज्मिड्स भी प्रेजेंट हो सकते हैं, विच मे बी ऑफ सेम टाइप और different type. So here you can see this is the chromosomal part, and here the blue color structure, it is the extra chromosomal part of the cell, which is double stranded molecule which is self replicating. It is known as plasmid. So what are the function of plasmid? Plasmids, it is called as ubiquitous in bacteria, means it is present everywhere in the bacterial cell. It may encode or it may consist the genetic information of that cell. So we'll study the function of plasmid one by one. First function is resistant to antibiotics. Now different types, anti means against and biotic means living organism. To kill or to remove or to reduce the number of bacteria, different types of antibiotics are given, and these antibiotics break the bacterial cell. So to provide the resistance to the bacteria against such antibiotics, plasmids play an important role. Then the second function of plasmid is bacteriocin production. These bacteria or these plasmid helps in the production of bacteriocin. Then it also produces different types of endotoxin or enterotoxins. The production of these toxin helps the bacteria to protect itself from the environment. Then fourth function of plasmid is enhancement of pathogenicity, means the pathogenicity or the ability to cause the infection can be increased with the help of plasmid. Next function of plasmid is it reduces the sensitivity to the mutagens. The different types of mutagens like chemicals or acids may cause the adverse effect to the genetic sequence of the bacteria. So the sensitivity to such material is reduced with the help of plasmids, as well as it degrades the complex organic molecules. The plasmid helps to break the complex organic molecule so that bacteria can easily utilize it or metabolize it and carry out various functions. So these are the role or the important functions of plasmids. These plasmids can be integrated or introduced in the different types of chromosomal DNA, which plays a important role in the genetic engineering. There are different types of plasmids or the plasmids are classified in the different types depending on their functions, which includes F plasmid. Now the F plasmid are the conjugative plasmid, which are also known as fertility factor. These plasmid codes the formation of sex pili, which favors the conjugation between F plus and F minus cell. Then the second type of plasmid is the R plasmid or the resistance plasmid. As we have seen the function of plasmid, it provides the antibiotic resistivity. So the R plasmid, it plays the important role as a resistant factor which codes the gene for the destruction or the modification of antibiotic and makes the bacteria resistant to the antibiotic. And then the col plasmid or the col plasmid, it plays an important role in the breakdown of the various complex materials. Then the next type is virulence plasmid. This virulence plasmid, it increases or enhances the pathogenicity of the bacterial cell. And the last type is metabolic plasmid, which helps to increase or enhance the metabolic property of the bacterial cell. So this was all about the extra chromosomal and chromosomal material present inside the bacterial cell. Thank you.